Good afternoon, everyone. Thank you for being on time. I realized this list of students are always the ones who are here right at the beginning of lessons. So kudos to you guys. We do have a very difficult lesson ahead of us today, and so I opt to teach it using the notes here, so that you can write it out along with me. The information are essentially the same as the ones that are shown in the PowerPoint slides. So I do encourage you to look through the PowerPoint slides afterwards, to kind of see the information from a different perspective, right? But I do believe writing it out is the best way to learn this difficult topic. Of course, there is the quiz that is scheduled for today, so please take the time to work on that as well. I see that no one has done the quiz just yet, which is fine. It's available to you until 11:59 tonight. So make sure you plan for that. So let's get started. Lecture 5, Metabolism. This is the last lecture of Unit 1. Your unit test is going to be next Thursday, right? And it will cover everything from Lecture 1 all the way to Lecture 5, as well as the lab, the Beetroot lab. Okay, we will do a review next class, but this week, we will focus on Lecture 5. So, previously we learned about metabolism, which are all the reactions that are going on in the body. We have the catabolism, which break things down, and in the process, it releases energy. And we can use that energy, we can capture that energy, and we can use it for various things. We can use it for transport, we can use it for anabolism, building things up, right? We can use it for movements, right? So the lesson today is focusing on one of the main catabolic reactions that happens in the body, which is cellular respiration. Okay, cellular respiration. You probably remember the equation for cellular respiration. I hope you remember, which is glucose plus oxygen, which produces carbon dioxide plus water, as well as energy in the form of ATP. So that's the word equation. I'm sure you learn in chemistry, you can represent an equation using words or using chemical formulas. So for glucose, it would be C6H12O6 plus 6O2. We have to balance the equation as well to make sure that we have the same number of atoms on both sides of the reaction. And for every glucose that we break down, we can extract enough energy to produce 36 ATP molecules. Okay. Now, let's take a look at this equation in a little bit more detail. Okay, so I'm going to leave some space. I'm going to draw something later in a bit, but I want to write an extra note here first. Okay, cellular respiration is a redox reaction. Which involves transfer of electrons from one reactant are the ingredients, right? One reactant to another. Okay, so that's what a redox reaction is. Something's going to be giving up electron, something's going to be picking up those electrons. There's a transfer of electron. We call those redox reaction. Now, the word redox is a combination of two words. We have the red part, read, R-E-D, that stands for reduction. Okay, reduction means gaining electrons. Okay, gaining electrons. So when something gains electrons, we call that reduction. You can remember GER, G-E-R. Gaining electron, we call it reduction. The ox here, that's oxidation. Oxidation, which is losing electrons. Loss of electron, losing electron, that's called oxidation. So you can remember Leo and GER. Okay, reduction, gaining electron, oxidation, loss of electrons. Okay, sorry, something's stuck in my glasses. So in cellular respiration, the glucose is the molecule that is going to be oxidized. Okay, so let's highlight it. Glucose is going to be losing electron and is going to be oxidized into carbon dioxide. Okay? We'll remove electrons from it, and once we're done removing electrons from it, it will become carbon dioxide. Those electrons has to go somewhere. They will go on to oxygen. Okay, they'll go on to oxygen, and they will be reduced into water. When they pick up those electrons, they will be converted into water. Now the transfer is not like a direct transfer. It's not like you take it from glucose and you put it onto oxygen right away. Those electrons has to pass through many steps in between, and eventually they will end up on the oxygen. Okay. Any questions so far? Nothing? All right. Can someone tell me where within the cell does cellular respiration take place? Please tell me where this
Basically in the mitochondria.
In the mitochondria, that's right, Justine. Part of it takes place in the mitochondria. Okay, so cellular respiration is actually broken down into 4 phases. Okay, so I'm gonna write them down here. Leave some space on the left, okay? Don't like if this is your etch of paper, don't write it here. Okay, leave some space here, and then we'll say step 1. Step 1 is glycolysis. Glyco means glucose. Lysis is to break. So glycolysis involves breaking down the glucose into 2 parts. And then we have step 2, pyruvate, oxidation. Just write them down first. Don't worry about what they mean. I'll tell you all about it in a bit. Krebs cycle. And then we have the electron transport chain. Okay, the ETC. So I'm going to add some stuff here in a bit. I'm going to add some stuff here in a bit. But just write them down first, and then I'll tell you where it happens, okay? Let me know if I'm going too fast, huh? All right, let's draw a cell here. Just like a tiny one. Okay, that's our cell. This is, of course, the cytoplasm. And what is this thing that I'm drawing? You should know. It's going to be on your quiz today. What's this thing? Anybody know what that is? We are in a bit of trouble if we don't know what that is.
Nucleus?
Okay, good try. Anyone else? Okay, maybe I'll have to draw better. What is this?
What is the thing that you drew inside, or what the arrow is?
The arrow. This whole thing is an organelle. What organelle is this?
Oh, that's mitochondria.
Mitochondria. That's right.
Yeah.
Right. Sorry, if it didn't look like it, but cellular respiration happens in the mitochondria, right? So step 1, glycolysis. That happens in the cytoplasm. Okay, step 1. Step 2 and step 3 happens right here. 2 plus 3. What's this place called? What's the innermost portion of the mitochondria? What do we call that, guys? Huh? Remind me.
The nucleolus.
You are confusing the mitochondria with the nucleus, Patrice. The inside of the nucleus is the nucleolus, but thank you for trying. Abnet said matrix, that's correct. This is the matrix. Okay, so step 2 and 3 happens in the matrix, and then step 4 happens right there. What are those foldings called? Do you guys remember? I don't think it's showing up too well here with my color. So step 1, 2, and 3, and then step 4 is right here on the foldings. What's the name of the foldings of the inner membrane? Yes, RJ, that's right. But it folds up into these special things. It has a name to it, right? Doesn't it? Starts with a C.
Is it Christe?
That is.
I don't know if that's how you.
No, that's exactly how you say it. Good job, Zoe. So glycolysis happens in the cytoplasm. Okay. The second and the third stage happens in the matrix, and the last one happens in the cristae. Okay. Again, if you have questions, please let me know. Okay, I'm sorry. I should have leave a little bit more space here between 3 and 4. I hope you have that space. Are we okay? Does anybody have any question? I'll post this again, and you probably should reorganize the notes after class anyways, to rewrite it, right? So that you can see it better. Okay? Just gonna move some of these things so I get more space. Sorry. All right. Let's go back to this picture here. In the first 3 steps, glycolysis, pyruvate oxidation, and Krebs cycle, we will focus on removing electrons from glucose. Right? Remember, this is a redox reaction. Something is going to be losing electrons, something else is going to be gaining it, right? And we will be removing it from the glucose. And that happens in the first 3 steps. Okay, so from the first 3 steps, we are gonna have electrons that are coming out. Okay, let's use an exciting color for the electron. That's not very exciting. I wanted to choose orange. There we go. That's the electron. And that's a high energy electron right there. Okay, high energy electron are being released from the first 3 steps. Okay. And what happens is you want to capture that high energy electron. We don't want to lose it. Right? We need to capture the energy as much as possible. So let's write down here. High energy electrons are released from step 1, 2, and 3. These electrons, I'm just going to do that for electron. These electrons are captured by electron, or sometimes they're called energy carriers. Okay. So there are 2 kinds of energy carrier that exist in the cell. We have NAD+, okay, and the other one is called FAD. Never mind what they stand for, it doesn't really matter. But pay attention here, okay? The NAD and the FAD, they will go to the first 3 steps. Let me try to draw this. They will pick up the electron. They pick up the electron, and then they will become NADH and FADH2. Now I'm skipping some of the details, right? It's already very complicated. So never mind where the H comes from. What you need to remember is before they pick up the electron, we call them NAD plus and FAD. After they pick up the electron, we call them NADH and FADH2. Okay, so basically I can write down here NAD plus and FAD are blank into NADH and FADH2 after capturing electrons from the first 3 steps. Okay. What should go into the blank here? What's the right word? Should it be oxidized into NADH and FADH2, or should it be reduced into NADH and FADH2? What goes in here? Please read the sentence and tell me. Yes, Blessing, I saw you unmute. You want to give that a try?
Reduced.
Reduced, absolutely correct, because they are picking up electron, right? Gaining electron. Gaining electron is reduction. They are reduced. Okay? So think of NAD plus and FAD as taxis, okay, or Uber. The taxi, when they have no passenger, the light on top is not lit up, right? Okay, so then the passenger, the electron can get on. Once they hop on, once they get into the car, it lights up, says occupied, right? So they become NADH and FADH2. So where will we take the electron? Okay, I'm sorry, I keep on moving this guy. Hoping you have enough space. So the NADH is going to drive the electron all the way to the ETC. Okay, same thing for the FADH2. They will drive it to the ETC. Okay, and then the electron will get off. Okay? The electron will get off onto the ETC, and then they will once again become FAD and NAD+. Okay? So basically, it's like a shuttle surface. Huh? They pick up the electron from the first 3 steps, drive them to ETC, goes back and pick up more, and then it just goes back and forth between the first 3 steps and the last step. Okay? So NADH and FADH2 deliver the electron to ETC, where they are oxidized into NAD plus and FAD once again. Okay? And where do they go? Well, they're just going to go back to the first 3 steps all day long. Pick up more, drive them to ETC. Go back, pick up more, drive them to ETC. Okay? Gonna pause for a moment here for you to catch up with the copying, and also please let me know if you have any questions about what I said. Nothing? Everybody's good? All right. So that's that. Now, what's the difference between FAD and NAD+, right? Well, it turns out they're very similar, but it's important to remember each NADH carries enough energy to produce 3 ATP molecules, whereas each FADH2 carries enough energy to produce 2 ATP molecules. Okay? Just think of them as different batteries. All right, like for example, like your calculator has those little circle cells battery, right? So those obviously don't carry as much energy compared to like the battery in your phone, right? So in the same way, NADH and FADH2, they're 2 different kinds of batteries, and one carries enough energy to produce 3 ATP. The other only has enough energy to produce 2 ATPs. Okay. So example, how many ATP molecules can be produced? Can be produced from 5 NADH and 3 FADH2. Okay. So why don't you try that out? You can use your calculator if you need to. Okay, and type the answer in the chat box when you have it, please. All right, I have 21. Anyone else? Anyone else?
I'm trying to copy.
I'm sorry?
I'm trying to get my answer.
Okay, sure. Okay, I have a few other 21s. So this is how we get it, right? 5 NADH. What do we multiply this number by? Well, each NADH carries enough energy to produce 3 ATP. So I'm going to multiply this by 3, right? So that gives you 15 ATP. Over here, we have 3 FADH2, which I can multiply by 2 because FADH2 only makes 2 ATP. So that's 6 ATP. If you add them up, we will get 21 ATP. Are we okay on this? If you see a question later on a test, do you know how to do the calculations? That's, the most important thing, right? Okay, feel free to ask if you're not sure. All right, moving on. So let's talk about ATP production. Okay. ATP molecules are created in 2 ways. All right. The name is really long. Just write down first and then I'll explain. Okay. First method is called substrate level phosphorylation. That's how you pronounce it, phosphorylation. Short form, we just call it SLP. Okay. See, one of the advantage of taking online classes, you don't have to spell long words like these, right? Just have to recognize it, right? Phosphorylation. What does it mean? Let's put a star here. To phosphorylate a molecule means to add a phosphate to it. That's all it means. It's like biology, they like to use complicated words for simple things. So if you add a phosphate to something, you phosphorylate it. Okay? So what is going on in substrate level phosphorylation? This is one of the 2 ways of making ATP in the cell, all right? So we're gonna need to have an enzyme. Okay? Everything needs an enzyme in the body, every reaction. Right? Enzyme helps catalyze reaction. Right? So that's an enzyme. And we have a donor molecule here. Okay? A donor molecule. So this donor molecule has a phosphate. It doesn't mind giving it away. It's like, you know what? Take my phosphate, no problem. I'll give it to you. Please take it. And then here we're gonna have ADP. Right? So essentially, this is what happens, right? Take a look first. Right? The enzyme will grab the phosphate from the donor, and then it will transfer it onto the ADP. What does ADP become after it picks up a phosphate?
ATP.
That's right, it becomes ATP, right? So that's how you generate ATP. That's one way anyways. Okay, so an enzyme, which is, a protein, right? Transfer a phosphate from a donor molecule onto ADP to produce ATP. Okay. So in this case, we are, like the enzyme is phosphorylating the ADP, right? It's putting a P onto ADP to make ATP. Okay, this is not a popular way, not I should say only about 11%, 10 to 11% of ATP molecules are produced this way. Okay, it happens in step 1 and 3. Okay, step 1 is over here. Step 1 is glycolysis, right? Step 3 is Krebs cycle. Okay, so this is not the main way of producing ATP. I know I keep on asking you like every 5 minutes, but seriously, questions? Anything you want to ask at all? Okay. Thank you, BB. The next step, the next method, right, of producing ATP, the common method, right, that would be called oxidative phosphorylation. Right, we just call it OP. I'll put some dots here if you want. OP, oxidative phosphorylation. Now this thing is very complicated. It would take me like half an hour to explain, which I will next class. But for now, I'm just gonna say, you remember those high energy electrons that we captured from step 1, 2, 3? Okay, so we're gonna be using those. Okay, using high energy electron to produce ATP in step 4. Okay, step 4 is ETC, huh? Electron transport chain. I'll just put it here as well. Glycolysis and pyruvate oxidation. There you go. Majority of ATP molecules are produced this way. Okay. That's all for now. As background information. That's all for now. Okay. Is pyruvate oxidation the same as Krebs cycle? Nope. Pyruvate oxidation is step. Oh, sorry, you caught my mistake. Thank you. Huh? Someone's paying attention. Very good. I'm sorry, I made a mistake here. Step 3 is Krebs cycle. Okay. Sorry. And thank you, Justine, for pointing that out. Step 3 is Krebs cycle. Step 2 is pyruvate oxidation. No ATP is produced from pyruvate oxidation. Thank you. All right. Are you guys up for some polls? I hope you are. Okay. These are all in the PowerPoint slides, but let's do this one. You can use your notes if you want and see if you can recall the information from memory. Excuse me. Please participate. Even if you don't know the answer, guess, right? Or, choose F to protest. It's like, I don't know. They are too difficult. Okay, I got half the class. Any more? Okay, well, the answers are kind of all over the place. Question asked, during cellular respiration, which molecule is oxidized? So if you took the notes, right? Look, glucose is oxidized, right? Glucose is the molecule that we will be ripping electrons from, right? Have I convinced you? Is glucose, like, it's literally right here, right? We shouldn't be choosing the products. Okay, we're only focusing on the reactant. Glucose is oxidized. Oxygen is being reduced. Okay. Hopefully that makes sense. How about this one? What's the right word? I am going to relaunch the poll. What's the right word? Anyone else? Last call. Again, I only have half the class. Where are the rest of you? Are you here? Or did you just join the class and went away for lunch? Oh, okay, yeah. All of a sudden, you guys showed up. That's great. All right, the correct answer is reduced, right? Reduced. Okay. NAD plus is blank into NADH when it accepts electron. Remember GER, right? Gaining electron is reduction. Losing electron is oxidation. The only choices is between B and C. Not that one, right? And not that one. We didn't even use those words, right? Look at the notes we took, right? I even color coded for you, right? See, NAD plus and FAD are reduced into NADH and FADH2. All right, so I hope those of you who did not choose C, hopefully, this makes sense to you. Are you guys like confused, or like were you guys just like missed this part of the notes? Like, I just want to know, right? Like, am I not being clear, or like you just missed that part? Because I mean, I thought it was pretty obvious what the answer was, right? Okay. You guys like to keep me in the dark. That's fine. Let's keep going then. All right. So now we're gonna focus on the actual process. Right? So 4 stages, right? 4 stages. We have the first step. First step is glycolysis. Glycolysis. Okay. Glyco is referring to glucose. Lysis is breaking. Okay. Remember, like if you put an animal cell in a hypotonic solution, it's gonna swell up and it could lyse, right? Lysis is gonna break. So glycolysis is to break down glucose into 2 pyruvate molecules. Okay. Sometimes pyruvate is also known as pyruvic acid. It doesn't matter. Okay, we can call it both. We're not gonna make a distinction here. Again, help me out, please. So I know that you guys are with me. How many carbon atoms do we have in glucose? How many carbon atoms do we have in glucose? 6 is correct. Thank you. So 6, we'll say 6 carbon. So if you split it into 2 pyruvate, how many carbon does each pyruvate have? You take glucose 6 and then you break it in half. How many carbon does each pyruvate has? That's right, 3. 3 carbons. Okay. Good. So keep those in mind. We're going to keep track of them. So glycolysis takes place in where? Which part of the cell does glycolysis take place in? Do you remember when we wrote it down earlier? Which part does glycolysis takes place in? You can check your notes, see if you took good notes. That's right, Amnet, thank you. Cytoplasm takes place in the cytoplasm. Okay. There are 10 steps in glycolysis. Before you throw your calculator at the screen and say 10 steps, that's too many. We don't have to memorize 10 steps, okay? I just want to tell you that there are 10 steps. Okay? And it is divided into 2 main phases. Okay, so phase 1. Okay, phase 1, which is the first 5 steps, right? That is the energy investment. Energy investment phase. Okay. The glucose doesn't really want to react. Okay, you might remember in lecture 2 we talk about activation energy, right? Activation energy is the energy you need to supply for the reaction to happen. So glucose is not going to react on its own. You need to prime it a little bit. You need to give it a little bit of charge, like charge it up a little bit with some energy, right? Before is going to react. And that's what the first 5 steps are. Okay, it's the energy investment phase, okay? 2 ATP molecules are used to energize glucose. Okay, think about like us waking up in the morning. It's like, oh, so tired. I don't want to do anything, right? So you have to eat a little bit of food, right? Drink some coffee, And now you feel energized. You're like, oh, let's tackle the day, right? So that's exactly what the energy investment phase is. You prepare the glucose, so it's going to have enough energy to go on and do all these reactions. All right, phase two. Phase two. This is the energy yielding. Yield means to produce, right? Energy yielding phase. Okay, so in energy yielding phase, we are going to produce 4 ATP molecules. Okay, and remember, there's a lot of things to remember, okay? But remember, we are trying to oxidize glucose, right? We're trying to rip electrons away from glucose. So in this phase, in the second phase, we will do just that. We will remove some electrons, but these are high energy electrons. We cannot let them escape. We must capture them. So we're going to bring in some NAD plus and capture them. So in addition to producing 4 ATP molecules, you will also produce 2 NADH. Okay. And at the end of it all, you get your pyruvate. Okay. So 2 questions here to ask yourself. It says we produce 4 ATP molecules, right? But really, how many did we actually produce net? What's the net production? Okay, I know we produce 4, but we pay some at the beginning as well, right? We charge it up, right? We use some ATP to charge up the glucose, right? So in total, how much did we actually make? How many did we actually make, I should say? What do you think? Do you guys understand my question?
Can you repeat the question, please?
Sure. Look, we used 2 ATP, right? Molecules to energize the glucose, and now we produce 4 ATPs. So when we take away the 2 that we produce, how many ATP did we actually make?
6.
Okay, interesting. Anyone else?
2.
2. Okay, and we also have 8. Oh my. Okay, let's think in terms of money, okay? You gave me $2. You gave me $2, okay? And then I take that $2 and then I do something to it. And then afterwards I make some money, okay? So I give you $4 back. How much money did you make? You gave me $2. I gave you $4 back. How much money did you make?
2.
That's right. Okay, not 6, not 8, not 3, right? Okay, that's exactly what's going on here. We used 2 ATP to energize the glucose. We spend 2 ATP. We spend $2. And afterwards, we get 4 back, right? We get 4 ATP back, right? So net production is just 2 ATP. Is that better? I'm sorry for confusing you guys, but like I hope the money thing made it better. Did it?
He does the money thing works for.
Okay, good. All right, yeah. Because the ATP is so abstract, right? Okay. Hopefully money is better. Okay, thank you. Another question. This one is harder, right? But the answer is there. We roll it down. Okay, I just want you to connect the notes, right? I will keep on repeating along the way to help you remember it. Okay, these ATPs, how are they being produced? Remember, there are 2 methods of making ATP in the cell, right? I don't want to say the name. I want you to look it up. Tell me, which method does this are these ATP produced by? Which method? First of all, find the names of the method and then look at what you wrote down and tell me which method is it.
Is this cellular respiration?
Well, okay, yeah, it's part of cellular respiration, but like which process, right? Blessing said, SLP, you got it. Okay, Vincent's asking so many difficult question today, right? Okay, so let's go back here. Right? ATP molecules are created in two ways. Number one, substrate level phosphorylation happens in step 1, glycolysis, and step 3, right? And then the other oxidative phosphorylation happens in step 4. Since we are talking about glycolysis, which is step 1, right? That means the ATPs are produced by SLP, substrate level phosphorylation. Okay? What that means is an enzyme is going to be here. It's going to transfer a phosphate from a donor to the ATP. All right? So over here, let me just write that down. Star, star. You should be using color. If everything is black and white, it's very hard for you to keep track, okay? So this is produced by substrate level phosphorylation. Okay? I got one more question, and then we're gonna move on. They capture the high energy electron, right? Electrons came from the glucose, right? We removed it from the glucose. We captured them. Now they become NADH. Where is this guy gonna go? NADH, where do they go, huh? What did we say earlier? It will become NAD plus again. Yeah, sure, eventually, but it has to drive the electron somewhere, right? It picked up the passenger now. Where are we gonna go? Where do we go?
Transport the electrons first, then become NAD again.
Yes, it's okay. So like the answer I'm looking for is this will go to ETC. All right. Let me just go back to show you. I'm just trying to relay everything for you, right? So that you can build upon it. I know it's a lot of information. I truly know that, and I'm sorry. But you know, that's what the ministry said you have to learn. So here we get the first 3 steps, releasing the electron. NAD plus picks it up, becomes NADH, and then it will drive it to the ETC at where it gets off, right? Okay. Think of them as like, you know, school buses. All right, you got the big school bus and then the smaller one, and then you pick up kids from different bus stops, okay? And then you drive them to the school. The school is the final destination. The kids get off, and then you can go back and pick up more kids, and you keep on doing this, right? Okay, so the ETC, the electron transport chain, that's the final destination for the electrons. Okay, they will get off at the ETC. And then at the ETC, we will use these electrons to make ATP. And how do we do it? We do it using this method, which I have not explained to you how it works yet. Right? Blessing, I'm sorry, it's my ugly writing probably. That says P, okay? P, OP, oxidative phosphorylation. Right? Well, it's not a D. Okay. So that is everything that's happening so far in the cytoplasm. All right. This picture is from your PowerPoint slide. You don't have to copy it right now. I am just gonna go through it with you one more time. Okay. So over here we have the glucose. Okay. Glucose has 6 carbons, each represented by these black circles. 6 carbons. In the energy investment phase, 2 ATPs are used to charge up the glucose, right? So that it will want to react, basically. And then we will remove some electrons which are captured by NAD plus, right? Okay, so NAD plus is reduced into NADH. And I asked you a few times already, where does this go? Goes to the final destination, the ETC. Okay, same thing over here. Right, it will go to the ETC. Goes to ETC. Right? And then we'll produce some ATP. Right? So these ATPs are produced by substrate level phosphorylation. Okay, and in the end, we get our pyruvate. Right? Pyruvic acid, same as pyruvate. Same thing. And each of them has 3 carbons in it. 3 carbons. Okay? That's all. Okay. This is the energy yielding phase, energy harvest phase. This means the same thing. It means that you're producing energy as opposed to spending, right? You've probably heard the phrase, it takes money to make money, right? Like if you want to invest money into a, at the bank, right? And then they will pay you interest, for example. That's what happens here. You invested some ATP, you're going to get some ATP back. Okay. Let's take a look at this question here. I am hopeful that you guys will get it right. How many net ATP molecules are produced? I will be heartbroken if you guys did not get this right. How many net ATP molecules are produced in glycolysis? Anyone else? Okay. Most of you got it right. The correct answer is 2, right? You spend 2 ATP in the investment phase, and then you get 4 back in the yielding phase. So total, 4 minus 2 gives you 2. This is another easy one. Go ahead.
Let's see the question.
Oh, sorry. It's asked how many carbon atoms are in each pyruvate. Is it 2, 3, 4, or 6? And again, I'm going to close the poll. Most of you who answered got it right. The correct answer is 3 carbons. Okay, glucose has 6 carbons. You split it into 2, 3, and 3. Okay. Now, next one is hard. I will just do it together with you. At the end of glycolysis, which of the following molecule has the most chemical energy? Now, is it NADH? Well, each NADH can produce up to 3 ATPs, right? So this is equivalent to 3 ATP molecules. Okay, so that means definitely not ATP, right? Because NADH already produced more ATP than 1 ATP. Carbon dioxide, we didn't even produce any carbon dioxide at glycolysis. That's out of the question. So which one has more chemical energy? Is it NADH or is it pyruvate? Well, at the end of glycolysis, we have only produced 2 ATPs, right? And in total, does anybody remember how many ATP can we produce from cellular respiration? From 1 glucose, what is the maximum number of ATP that can be produced? Look at the notes.
36.
36, exactly, right? So after glycolysis, we have only produced 2 out of the 36. That means there are still 34 that are locked up in the pyruvate, right? So this will go on and create much more ATP, right? From step 2, step 3, step 4, right? So most of the chemical energy is still locked in pyruvate at the end of glycolysis. Okay. All right, we're going to take a break. We'll come back at 1:37. We will be using the study guide a little bit. So, if you haven't downloaded a copy, please go ahead and do that. And I'll see you in 5 minutes, all right? Alrighty, let's keep going. So, before the break, we were just talking about glycolysis, how we invested some energy into the glucose, and then we are able to harness and basically get more energy, more than what we have invested, right? And at the end of it, we have produced the pyruvate. Okay, so we have 2 pyruvate molecules from the glucose. So let's take a look at the fate of these 2 pyruvate molecule. Okay, step 2, pyruvate oxidation. Okay, pyruvate oxidation. So we had the glucose from the beginning, and I'm just drawing a continuation of what we had there. We have the pyruvate, 2 pyruvate. Right? So 6 carbon in glucose, 3 carbon in the pyruvate. Nothing is new so far. Can somebody tell me where will these pyruvate go? Okay. I'll write down here. The 2 pyruvate molecules produced in glycolysis move to where in the cell? What did we say earlier? We drew a picture. We put the 1, 2, 3, 4. Where are the pyruvate gonna go?
Is it mitochondria?
That's right. It's gonna go to the mitochondria. Specifically, which part of the mitochondria, though?
Matrix.
The matrix. Excellent. So the 2 pyruvate molecules produced in glycolysis move into the matrix of the mitochondria. Okay. Again, ION is just singular, IA is plural. In the matrix, 3 things are gonna happen to our pyruvate. Okay? 3 things are gonna happen to them. Number 1, we will remove a carbon dioxide. Okay. So step 1, remove CO2. As the name of this step implies, pyruvate oxidation, we are going to be oxidizing pyruvate. We're going to be removing more electrons from it. Every time we remove electron, of course, we have to pick them up. We have to capture them with energy carrier. So an NAD plus will come along, the taxi will come along, school bus, whatever you call it, will come along, pick up the electron, and then it's going to become NADH. And you should know by now, because I've been repeating it all afternoon, this NADH is going to take the electron to the ETC and let the electron get off, right? Now, if you're still with me, listen for a second, stop copying, right? By this point, the molecule is kind of running low in energy. Okay, we produced it some ATP earlier. We removed quite a bit of electron, and now the molecule is out of juice. Right? It's like doesn't really want to react anymore. Right? It's again like back to that breakfast analogy, right? You wake up, you're tired, you don't want to do anything. You eat the breakfast, you drink some coffee, you're ready to go. Okay, but then it's noontime now. You used up all those energy, right? You feel tired again. You need a little bit of pick me up, right? You need to have a snack, you need to have lunch so that you can keep going. And that's exactly what's happening here. The molecule is not really willing to react further, and so we're gonna energize it. Okay, we're gonna energize it by adding something called coenzyme A. Okay, so three things happen. First step, remove carbon dioxide. Step two, right, produce NADH. That's just capturing the electron. And number three, coenzyme. Coenzyme A energizes the molecule so that it will react further, right? So that it would want to proceed, right, with all these other extra reactions. So in the end, we have this thing called acetyl-CoA. Let me just shorten the arrow so I have space. Okay, in the end, we have this thing called acetyl-CoA. Now let's do some more math here. Can somebody tell me how many carbons do we have in the acetyl-CoA? How many carbons? Anybody? Don't think too hard. It's pretty straightforward. Okay, Justine says still 3. Anyone else? Do you agree it's 3? Do you think it's something else? Two. Why is that 2, Abnet? I agree with you, but yeah, go ahead.
Because
Mhm.
it's like a crucial intermediate in cellular respiration.
Okay. Abnet, you want to explain your reasoning or no? Or anyone else? Yes, because of the CO2, right? You have 3 carbon dioxide here. Sorry, you have 3 carbons here. We remove the carbon dioxide. There is a carbon in carbon dioxide, right? So 3 minus 1 gives you 2. Does that make sense? Okay. All right. So this happens to both of the pyruvate. Okay. This happens to both of the pyruvate. And that means for every glucose that we get, we end up having 2 acetyl-CoA. All right. I'm gonna pause for a second, let you catch up with the notes. And of course, ask questions if you have any. Are we good? Can we go on? Give me some feedback here, guys. What's going on? All right. So that's the professional drawing. Okay. And there is some other intermediate here called acetic acid. Never mind that. You don't really have to know that. Okay. But same thing, right? Remove the carbon dioxide, pick up the electrons, add the coenzyme A to charge it up, and then we end up having the coenzyme A with 2 carbons in it. Okay. We call this acetyl-CoA. Acetyl-CoA. All right. Let's go to our table here. I have to make a copy of it. Give me a second. There we go. All right. So if you have your study guide, it's just on the first page. If you don't have it, that's fine. You can copy the table. Okay. So let's fill it in. Stages in cellular respiration. First step is glycolysis. Right? Glycolysis. You knew that already. And that, of course, takes place in the cytoplasm. The main things, the main products of glycolysis include 2-pyruvate, right? 3 carbons each. 2 net ATP. Remember, we invested 2, and then we got back 4, right? So net is just 2 ATP. And also 2 NADH. And I know I said this before, but let's just put a pink star here one more time. That is produced by, as Blessing answered this earlier, this session is SLP, substrate level phosphorylation. Okay. The enzyme's gonna take the phosphate from a donor, transfer it onto ATP. Second stage of cellular respiration, pyruvate. Pyruvate oxidation. And that happens in the matrix. Right? What do we get? We have 2 carbon dioxide, 2 NADH, and 2 pyruvate. Right? This is per glucose. Right? Because each glucose makes 2 pyruvate, and each pyruvate will be releasing its own carbon dioxide, right? So there are a total of 2 carbon dioxides, 2 NADH. And oh, sorry, this is a wrong thing here. Then and 2 acetyl-CoA. Okay. That's all. We are almost there. Just gonna do one more stage, and then we'll call it a day. All right, next step. Next step is called the Krebs cycle, right? Step 3. Krebs cycle is the person's name. Person who I don't remember, like either discover it or contributes to finding it. Okay. It's also called the citric acid cycle. Okay. Both is okay. All right, so let's just do another mini recap. Repetition is the key to remembering these things. Okay, so glucose is going to split into pyruvate. This is called glycolysis, right? I hope you know it by now. And then these pyruvate will go into the matrix where you will do the second step. Okay, so in the second step, remove carbon dioxide, produce NADH, and we recharge it with coenzyme A. So we end up having acetyl-CoA. Which is 2 carbons, right? We lost a carbon because we removed the carbon dioxide. So this is pyruvate oxidation. Okay. Now we are in the matrix right now. And where does Krebs cycle take place in? Do you remember? Where does Krebs cycle take place in?
Matrix.
Also the matrix, that's right. So they don't have to go very far. They are already in the matrix. All right. Now, this is a little bit weird, but try to stay with me. In the matrix, there is a molecule called oxaloacetate. It was already there, okay? And it's 4 carbon base. So these two will combine, right? And then you get citric acid. 2 plus 4 is 6. Okay, so that's why it's called the Krebs cycle is also called the citric acid cycle, because the first product in this stage is the citric acid, right? And then there will be multiple steps in between. But ultimately, because it's a cycle, you will regenerate the oxaloacetate. Okay, so in the process, a couple of things comes out. Right, we have 2 carbon dioxide. Okay, we have 3 NADH. Because electrons are being given off, right? So we have to capture them. And we're going to have 1 FADH. And also, we're going to have an ATP. And of course, this ATP is by SLP, substrate level phosphorylation. Okay? We wrote it down earlier. Substrate level phosphorylation happens in step 1, glycolysis, and step 3, Krebs cycle. Okay. So those are the product. Now you can see we have 2 carbon dioxides here, right? Okay. And because the glucose produces 2 acetyl-CoA, each of the acetyl-CoA is going to undergo the exact same thing. Okay. I'll give you a moment to draw this in a bit, but look, 2 carbon dioxide here, 2 carbon dioxide, that's 4 carbon dioxide. Let's go back to here. We have 4 from Krebs cycle, 5 and 6, right? So that accounts for the 6 carbon dioxide here, right? Okay. Let me write that down for you. By the end of the Krebs cycle, glucose is completely broken down into CO2. Okay. So from the 6 carbon, you take away the 2 carbon in carbon dioxide. That's how you get back to 4, right? And then you can combine with another acetyl-CoA, and then it's a cycle. Okay, so a couple of other things to pay attention to. Each cycle produces 3 NADH, 1 FADH2. I'm sorry, I'm missing the 2 here. And 1 ATP. Okay. However, each glucose produces 2 acetyl-CoA. So each glucose produces 6 NADH, right? 3 plus 3 is 6. 2 FADH2 and 2 ATP in Krebs cycle. Okay. So here's a question for you to think about. What is the maximum number of ATP that can be produced with 5 turns of Krebs cycle? Okay. Not just these ATP directly, right? But you remember these NADH and FADH2, they can make ATP as well, right? So please try that and type the answer in the box when you have it. What do you guys think? All right, I got a 30. Anyone else? 10. Anyone else? Okay, also 10. 5. All right. I don't know if you guys heard me, but I said, right, like not just the ATP that are produced directly, right? These NADH and FADH2 will go on and make ATP as well, right? So together, accounting everything, how many ATPs are we going to be able to get? Okay, maybe we'll do this one together. Look, one cycle, one turn, right? One turn, what do we have? According to the notes, one cycle produces 3 NADH, 1 FADH2, and 1 ATP, right? So tell me, what should I multiply this by? How many ATP can we make from 1 NADH? Do you guys remember? 3, that's right. So I'm gonna multiply by 3. 3 times 3 is 9 ATP. Okay, how about here? What should I multiply FADH2 by? Very good. I hope the rest of you agrees. Right? So that's 2 ATP. And ATP here is just ATP, right? Like I don't have to times anything. You just count it as one. One is one. So I add these up. I add them up. 9 plus 2 plus 1 is 12 per cycle, per turn, right? So the question is asking you to do 5 turns. So 12 times 5 is 60 ATP. Does anyone have any question about that? All right. Let's put a twist to this. What is the maximum number of ATP that can be produced from Krebs cycle if 5 glucose molecule are metabolized? Okay, give that a try. Don't worry about copying the question now. I'll post the notes, right, after class. Focus on solving the problem now. Anyone? Zero? Interesting. Why is that, Justin? Justine?
I don't know, sir. Like, I think was metabolized, break it down, so didn't make anything. But I know, have no clue.
Oh, okay. This is not a trick question like that. uh, Interesting interpretation, though. To heaps at 15. Okay, anyone else? You guys can do it. Come on, please. Okay, let's think about this. How many times does the Krebs cycle turn for 1 glucose? If we break down 1 glucose, how many times does the Krebs cycle spin? Anyone? Two times. Thank you, Blessing. That's correct. If you didn't know that, let's take a look here. We started with 1 glucose. Each glucose is broken down into pyruvate. Each pyruvate is converted into acetyl-CoA, and each acetyl-CoA will spin in the Krebs cycle, right? So that's 1 spin and 2 spins. All right. Now, let's move on to the next part of the question. We know that 1 glucose corresponds to 2 turns, 2 spins. So now we have 5 glucose. How many spin is that? How many turns? For 5 glucose, how many turns? Hopefully you will know it's 10 turns. Thank you, 10 turns. Okay. How many ATP per turn? Well, we worked it out last time. Per cycle, per turn is 12 ATP. So we take 12 ATP per cycle per turn, same thing. Multiply by 10 turns, 12 times 10, 120 ATP. Okay? Please familiarize yourself with these questions. They will show up on your tests. So just to wrap things up, let's go back to our table here. You know, we have the last step here, which is the Krebs cycle. And it also happens in the matrix, right? So in terms of product, we have 4 carbon dioxide, 6 NADH, 2 FADH2, and 2 ATP. So 2 carbon dioxide here, 4 carbon dioxide here. That makes up the 6 carbon dioxide, right? In cellular respiration. By the end of the Krebs cycle, the glucose is completely gone. Gonna put a star here as well. These 2 ATPs, along with the other 2, are produced by SLP. Right, so that's 4 ATP in total, right? 4 ATP in total. Out of a total of 36, right? So if you do 4 divided by 36, it's 11.1%, right? It's not nothing, but it's not a majority, right? So a small amount of ATP is produced using SLP. A majority of the ATP are actually produced in step 4 using the oxidative phosphorylation. And that is what we will be learning on Thursday. That takes like half an hour to explain. So like I don't want to start now. We're not going to be able to finish, right? So that's it for today. It's a very complicated lesson. All right. So I know you have the quiz to worry about. So focus on that first. Good luck with it. But afterwards, you should go through this material, especially before coming to the next class. You want to go through this thing. Maybe watch the recording again. You know, and then we will finish it by Thursday. Okay. Thank you for coming. Take care, guys. See you Thursday. Bye for now.
Excuse me, sir.
Yeah, go ahead.